Growth and differentiation of the epidermis. Introduction. The epidermis must maintain a largely impenetrable barrier to the outside world over the course of a lifetime. Simultaneously, it rapidly turns over, replacing itself every 2 weeks, and retains the ability to heal wounds. To accomplish these divergent roles, the epidermis has a highly regulated proliferation and differentiation program. Progenitor cells within the basal layer of the epidermis have a very high capacity for proliferation and self-renewal, whereas the major role for differentiated cells is generation of a barrier. Epidermal growth includes epidermal stem cells, epidermal stem cell niche, and epidermal proliferation. Epidermal stem cells. Progenitor cells that can both renew themselves and give rise to differentiated progeny over an extended time period. Isolated basal cells express varying levels of a number of genes. Cells with high expression of these genes have higher self-renewing capacity in vitro and tend to form clusters in intact skin. Epidermal stem cell niche. The tissue microenvironment in which stem cells normally exist is called their niche. Niches are continually being more thoroughly defined and include factors such as direct cell-cell and cell matrix communication, soluble secreted factors, and local tissue mechanical properties. They are located dispersed throughout the basal layer, or at either the tops or bottoms of reedy ridges. Part of this may reflect regional differences in skin architecture. The progenitor cells in the basal layer largely produce differentiated progeny that form in columns directly above them. Epidermal proliferation. The epidermis requires continuous proliferation for both homeostatic turnover and wound healing. It is thus important to understand whether there are specific kinds of cells that are amplified or hyperproliferative in response to different stimuli, whether cells are inhibited from differentiating, and what molecular pathways are driving the overproliferation. These include endothelial signals, inflammatory signals, and extracellular matrix rigidity. Many of the major developmental signaling pathways to regulate epidermal proliferation and major downstream targets are involved. Structural markers of epidermal differentiation. Basal cells are defined by basement membrane attachment, the presence of hemidesmosomes, and the expression of keratins 5 and 14. Keratin 1 and 10, in contrast, mark all differentiated cell layers, whereas loricrin and filaggrin are expressed in upper spinous and granular cells. All cells contain desmosomes, though the makeup of desmosomes changes, with desmoglein 3, DSG3, and plakophilin 2, PKP2, being highly expressed in basal cells, whereas DSG1 and PKP1 are highly expressed in differentiated epidermis. Tight junctions and keratohyalin granules mark the granular cells. Epidermal differentiation includes basal cells, spinous cells, granular cells, and stratum corneum. Basal cells. Basal progenitor cells are characterized by their mitotic activity, which is necessary for normal epidermal turnover and wound healing, marked by the expression of keratins 5 and 14. Keratins are members of the intermediate filament family of proteins and are obligate heterodimers that assemble to form polymers in the cell. This large family of proteins shows tissue and cell type specific expression. The keratin filaments stabilize both cell-cell adhesions called desmosomes and hemidesmosomes, providing mechanical anchoring roles. In addition to hemidesmosomes, basal keratinocytes are attached to the underlying substratum by focal adhesions, which bind to filamentous actin in the cell and play many roles, including promoting survival and proliferation of keratinocytes. All keratinocytes, including basal keratinocytes, have cell-cell adhesions called adherens junctions, which are actin-based structures. Adherens junctions components have diverse functions, from regulating cellular architecture to growth control and inflammation. Basal keratinocytes give rise to spinous cells during epidermal homeostasis. The production of cells in the suprabasal cell layers is driven by mitotic spindle reorientation and asymmetric cell divisions during embryonic development. Spinous cells. Basal to spinous differentiation is a highly regulated transition. Cells switch from a mitotic, keratin 5/14 expressing type to a postmitotic state characterized by the expression of keratins 1 and 10. There is an upregulation of desmosomes in these cells that gives them a spiny appearance in histologic sections. The composition of desmosomes also changes on differentiation. Although basal cells are high in the desmosomal cadherin, desmoglein 3, the levels of this protein decrease through differentiation, whereas desmoglein 1 is upregulated. Granular cells. The identifying feature of these cells is keratohyalin granules. These granules are composed of keratins, profilagrin and loricrin, and other proteins that make up the bulk of the cornified envelopes. Filagrin and loricrin are also commonly used markers for these cell layers. Upon secretion, keratins and loricrin as well as other proteins, including small proline-rich proteins, become highly cross-linked by transglutaminase to the plasma membrane to generate cornified envelopes. Profilagrin is eventually metabolized into amino acids and pyrrolidone carboxylic acid and urocanic acid, sometimes referred to as natural moisturizing factor NMF, to provide hydration and UV protective functions. Many of the genes required for formation of the terminal differentiated epidermis are contained in the epidermal differentiation complex, a region on chromosome 1q21. These include loricrin, involucrin, small proline-rich proteins, and late cornified envelope proteins, to name a few. Tight junctions are cell-cell adhesions that restrict the flow of fluids and ions and act as a barrier to membrane diffusion. These structures form specifically in the granular layer, thus allowing diffusion in the intercellular spaces of live cells within the lower layers of the epidermis. At the core of tight junctions are claudins. The last function of granular cells is to die. The resulting cornified envelopes are not living cells, and thus nuclear and cytoplasmic contents must be eliminated. This is thought to be a modified form of programmed cell death. Stratum corneum. Cornified envelopes are the products of terminal differentiation in the epidermis. They are acellular and anuclear structures. The core consists of keratins surrounded by highly cross-linked networks of proteins, especially loricrin. Specialized lipids surround these, rich as ceramides that are also cross-linked by transglutaminases. The cross-linking of proteins and the presence of specialized lipids result in mechanical stability and relative impermeability of the epidermis. While the stratum corneum forms an outside-in barrier, the tight junctions form an inside-out barrier and thus collaborate to form an effective barrier to the external environment. Epidermal differentiation regulation. Transcriptional regulators. P63 controls epidermal specification, but it also plays an important role in maintaining basal cell fate or proliferation and induction of differentiation. 21 Direct targets of P63 include both structural proteins and other transcription factors that regulate differentiation. KLF4, another transcription factor, upregulates the expression of a number of lipid-modifying enzymes and structural proteins important for cornified envelope production. Notch signaling is a commitment factor for transition off basal cells to spinous cells. Transcription factors of the grainy head-like GRHL are also key promoters of epidermal differentiation. Epigenetic regulators. Epigenetic regulators control the organization and/or accessibility of chromatin for transcriptional regulation. In the epidermis, differentiation is driven by coincident expression of transcription factors and chromatin states. The process includes DNA methylation, histone modification, chromatin remodelers, and post-transcriptional regulation. Regulators of epidermal differentiation. Transcription factors and epigenetic regulators of epidermal proliferation and differentiation are indicated. The position of the labels corresponds to the area of the epidermis where the factor is first expressed. Although direct interactions and regulations are known between some of these factors, this is both complex and incompletely understood. The text provides additional insight into cross-regulation and structural target genes. Conclusion. As the epidermis needs to provide an essential barrier to the outside world and to continuously renew itself, the decisions of proliferation and differentiation are highly regulated. Further understanding of the physiologic and pathologic control of proliferation and differentiation decisions is expected to lead to better treatments. In addition, the incredible regenerative capacity of stem or progenitor cells within the basal layer has already been clinically harnessed to treat burns and genetic skin conditions.